Welcome to Brain Ponderings. I'm your host, Mark Matson. My guest today is Darren Baker. He's professor of biochemistry and molecular biology and pediatrics at Mayo Clinic in Rochester, Minnesota. He's expert on regulation of cell cycle and the process called cell senescence, which is what I'll talk with him about today. He's done a lot of pioneering work somewhat on molecular mechanism of senescence, but he's got really interested in brain aging, neurodegenerative disorders, and has some exciting data suggesting cell senescence does occur with brain aging and Alzheimer's disease and may play a role in the pathogenesis. And so we'll talk about that as well as about emerging therapeutic approaches based on targeting these senescent cells and removing them or otherwise affecting them. So welcome, Darren.
Thank you very much for having me, Mark.
And are you from Minnesota originally?
I am originally from Minnesota. My hometown is about 3.5 hours away from Rochester, Minnesota. So I knew Rochester, Minnesota, or at least what I knew of Rochester was the Mayo Clinic, the clinic part of what is being offered here. Because I was actually a patient here when I was in second grade. I was really quite sick when I was a kid. So when I was sick, my parents had options about where they could take me for treatment. And of course, they were going to take me to Mayo Clinic instead of my local hospital that was there. And so I was a patient in St. Mary's Hospital for quite a long time when I was in second grade. So I was again, quite a sick kid. I had some kidney issues that were then essentially alleviated. So now I've been really healthy since then. And so I knew Rochester and Mayo Clinic for that perspective, but it wasn't until I was graduating from my undergraduate, which was at the University of Minnesota, that I was walking around and there was a career fair and there was a big booth that was talking about Mayo Clinic research. And I was thinking, well, I kind of want to do some research versus what I had worked on in my undergraduate was things about looking at herpetology studies, looking at chorus frogs and salamanders was what I had worked on in my undergraduate. And thinking that if I wanted to go to medical school, what better place could I get for having research experience would be than Mayo Clinic. So that was essentially my entry into why I became interested in coming here in the first place.
Okay, and so, but you didn't come directly to Mayo, right? You went, did you go to Europe for your PhD?
So my path is a little bit confusing if you look at it. So my path, I did my undergraduate at the University of Minnesota. I came to Mayo as a research tech originally with the intention of being here, getting research experience and going to medical school. During the time that I was doing my research, which we'll talk about, I did all of the parts about medical school of doing the MCAT, doing the admissions process, getting into medical school. And I deferred my matriculation for a year before I was going to start. And that fall, when I would have been starting medical school, which was at the Medical College of Wisconsin in Milwaukee, was when my first son was being born. And so I kind of made a judgment call at that point in time of should I go to medical school and do this thing with a young developing family, or do I really enjoy what I was doing from the research side? And I chose to really stay in the research side. And so then kind of have changed trajectory from that point in time. At the time, Mayo, I was able to enroll in the employee master's program from Mayo Clinic. So my master's degree is from Mayo in the context of tumor biology, which we'll talk about where tumor biology is relevant for senescent cell biology and how we got into it. That's what I did first for my master's degree. And my PhD work is from the Netherlands, Radboud University in Nijmegen is where my PhD was. So the work for PhDs in the Netherlands is based much more on your scientific contributions to the field, meaning publications, more so than having a particular thesis project that you propose, you go through, and you end up hopefully with a paper at the end of it. At least that's typically what our graduate school here of Mayo is. In the Netherlands, it's much more of a comprehensive, you have to have kind of a collection of stories around a topic that you can then apply for. And so because of my primary mentor being originally from the Netherlands, having still associations with Radboud University. In fact, my mentor from the Netherlands side was my primary mentor's mentor. So like the scientific grandfather, if you will. And so that's kind of the way that path had gone.
Oh, very interesting. And early on then you got interested in cancer.
Mm-hmm.
And of course, cancer cells divide out of control.
Correct.
And they're generally resistant to a process called apoptosis, which is a way that the body naturally removes cells that may become cancerous. So can you talk a little bit about that work? Because that really led you to senescence. You're studying cell cycle regulation.
Exactly. And so what I started in my research career in the laboratory of Jan van Dersen, that Jan was very interested in a concept about aneuploidization, meaning an abnormal complement of chromosomes. A German person named Theodor Boveri, about 120 plus years ago now, made a recognition of looking at human cancer cells. When he was looking at them, he saw that they were very abnormal for the number of chromosomes that were in those cancers. You know, typically you and I are walking around with 46 chromosomes. Having just one extra copy of one chromosome, for example, in Down syndrome, most of those patients have one extra copy of chromosome 21. That's the only abnormality that those people have from a cellular basis, but it's very stable, meaning that all of the cells of the body have the same thing. But there's pretty significant ramifications for the overall fitness of those individuals by having just that one extra copy. What Theodor Boveri had noticed when he was looking at human cancers was not that it was plus minus one or two chromosomes. They were crazy spreads of 60, 70 chromosomes. So very unusual. And he made a postulation at the time of maybe chromosome instability, where you have these random gains and losses of chromosomes, is sufficient to promote the early stages of tumor genesis, where you maybe have a chromosome gain that has, at the time it wasn't known about this, but having a lot of tumor promoting effects and oncogenic activity. You maybe had a chromosome loss that had a lot of tumor suppressive activity, or the various protein imbalances that come along with that, that those would be sufficient to kind of stimulate the process of tumor genesis. So what Jan was trying to do at the time was we were trying to convince cells to go into these abnormal segregation of their chromosomes, or at least having aneuploidy. And the way that we thought we would do that is by really manipulating a particular point in the cell cycle. You know about the cell cycle and kind of the idea about cancer, was that we were looking specifically in mitosis. If you've looked and seen live cell imaging videos of cells as they're going through mitosis, you have this very beautiful condensation of chromosomes. They all align in the metaphase plate, and there's a very synchronous, beautiful division into two identical daughter cells. Lovely, lovely cellular process. That doesn't just happen by chance or accident. There's a whole bunch of proteins that play a role in making sure that this is maintained in a very high fidelitous manner. Something that's called the spindle assembly checkpoint, or the mitotic checkpoint, makes sure that all of your chromosomes have the proper tension and will then segregate into two identical daughter cells. There's probably about 30 or so proteins that play a role in that process. And so what we started doing, I'm a mouse biologist. I run also the transgenic and knockout core facility of the Mayo Clinic. So any mouse that you want to make, we can definitely do that. But what we were doing was we were then manipulating different players that were part of this surveillance machinery to try to convince cells to missegregate, thinking that then we are going to test the hypothesis that chromosome segregation alone is sufficient to promote tumor genesis. Okay? We made probably about 15 different models that were in that space. Most of them, if you really stimulate or challenge them with a carcinogen especially, they do demonstrate a clear predisposition to cancer, suggesting that in situations where now you're maybe having oncogenic mutations or tumor suppressor loss, the addition of now chromosome instability on top of that is really stimulatory for cancer. There was one model that was very unusual in that it didn't really have a very strong spontaneous cancer phenotype. We did drive cancer if we treated it with a carcinogen, but these mice were dying really, really rapidly. And so instead of like throwing that mouse in the bin because it wasn't a very good cancer model and we were tumor biologists, I became very interested in why is this mouse so sick? And so that's why kind of we got into where this mouse has pathology. We were trying to, at the time in the early 2000s when we started this work, we were then noticing it was not uncommon in high profile journals to see DNA mutation mouse models or mitochondrial mutation mouse models or telomere deficient mouse models, things that are very prematurely aged having pathology in the pages of Science, Nature, and Cell. And so looking at those, they talked about these mice having progeroid or premature aging syndromes, where maybe they had hair graying or hair loss or all kinds of other phenotype. And so by reading those papers, we started then looking a little bit more at our mice again and saying, well, they don't have hair graying or hair loss so much, but all of my mice have cataracts by 2 months of age. I have elderly grandparents. Cataract surgery is something that Mayo Clinic does with a high degree of regularity around here. So I know that that's very much an age-related condition. And so we started then really examining this mouse model to say, well, does it have premature aging syndromes? And we found a number of different tissues that develop these pathologies. But when we tried to jam it underneath the known pillars of the time about why a mouse would have a premature aging phenotype because of maybe DNA damage that's unresolved, or short telomeres, or excessive mitochondrial damage, things like that, it never fit underneath any of those pillars, which is why we really kept digging into what is going on with this mouse. And we found in certain tissues, there was a really high acquisition of senescent cells, which we characterized at the time of being primarily P16 expressing, but then also beta-galactosidase positive, a couple different measures that we'll talk about in the context of senescent cell biology, that those mice seem to have some of those features early on.
Darren, could you describe the phenotype or what a senescent cell distinguishes a senescent cell from a normal cell?
Yeah. We're still working on that question, even though it's something that has been known for quite a long time. The concept about cellular senescence, so the Theodore Boveri idea about aneuploidization driving tumor was a 120-year-old hypothesis that we were kind of testing. The idea about senescence, that's much more recent. That was in the 1960s, was when that was about the idea about noticing that cells in culture, at least human fibroblasts, up until that point in time, the prevailing thought was that cells were immortal, that if you brought human cells into culture, you can continuously proliferate them pretty much forever, which was somewhat unexpected. That's a thing that you might expect in the context of tumors or cancer cells, but normal cells, you maybe not necessarily expect that. So it was really Len Hayflick that was the person that had done a lot of the work in the laboratory of Paul Moorhead in the 1960s to really demonstrate that human fibroblasts, by now having the right type of media, by passaging over time, that you would see that there is a phase where they will proliferate. But then after a certain number of divisions, which seems to be really well programmed, it goes into a state where they're no longer capable of division. Those cells, the apoptosis that you talked about, the cell death process, don't seem to be activated in senescent cells. In fact, there's a lot of pathways that seemingly are on in encouraging those cells to stay in a survival state, which is where some of the therapeutics are coming into to now try to convince these cells that have turned on these pathways to be alive to now go into a death process. But they become in a state where they're very metabolically active. They're still secreting, and what we've found since that point in time is they start secreting a variety of factors. They really change their behavior completely. So the concept about senescent cells, and when we're talking about your Brain Ponderings podcast related to neurons, neurons by definition are an irreversibly growth arrested cell, which is one of the things that people talk about senescence. But I don't really think that that's alone sufficient to really call a cell senescent. What I think about senescence is I more think about the behavior changes that are now taking place in those cells. They're starting to do things that they haven't historically done, maybe becoming very non-functional in relevance to what they've been doing historically as well. And part of that then is the acquisition of a variety of different features. Some of them might be a very pro-inflammatory state, where they're secreting a number of molecules that can influence other cells in the environment. The concept about senescence is, largely, if you look at much of our work, it's essentially senescence equals bad, is kind of the simple answer. But in essence, we have this process for a variety of purposes. And in a short stage, kind of what we define more in the acute stage, there seems to be a very important role of senescence in terms of activation of immune responses, having benefits for wound healing responses, having other things that kind of play a very transient role for benefit of having these cells that maybe are now having the secretion of these factors. One of the places
even in normal development of different organs, right? There's evidence that senescence is kind of a normal.
Correct, correct. And I was just going to get to that in terms of the in developmental process. I think we're still, it was surprising to find senescence there, because in development, you're having a lot of cells that are dividing very rapidly. And now why would you trigger this irreversible or this long-term stable growth arrested state? But one of the conditions that I like to talk about, because I think it's a good example, is that there seems to be senescent cells that will accumulate at the tips of the digits that are developing within animals. When they do that, they now start to secrete a variety of factors, things like the matrix metalloproteinases that are destroying extracellular matrix, such that then you get actual separation of the digits of the hand. So it plays a very important programming effect of that. If you don't have the ability to engage senescence, you see that this doesn't happen with the same type of regularity, versus if senescent cells are here, they secrete the factors that are necessary for then having the separation of what was the developing digits. Just as one of the examples for where developmental senescence seems to be playing a beneficial role.
There's a certain kind of Labrador retriever dog, Chesapeake Bay Retriever, and they swim really good. They have webbed feet. I'm wondering if they have like a mutation in a metalloprotease gene or something, or if there's a role
or a bypass. I'm not sure, because there's all kinds of animals
you
that
need
would
to
have
look
that.
at senescence markers in the feet of those dogs.
With development. Yep. No, possibly.
Okay, but anyway, so senescence, it's important for normal development, can suppress tumors in a way, right?
From a
And
cell
then maybe
intrinsic
wound
effect.
healing, some other things.
Correct. From a cell intrinsic effect, and this is where we think about senescence, maybe that's occurring in the context of that Bubber1 hypomorphic mouse model, is that their cells are becoming senescent for a particular reason. So this mutant mouse model that we made, it was having chromosome missegregation, but we had other models that were having chromosome missegregation that had no senescence effect. So it was something really unique about that particular gene and that protein, about why are those cells now becoming sensitive to really becoming senescent versus maybe others that we mutated did not have that same effect. From a cell intrinsic mechanism, you can think of senescence being really a beneficial anti-cancer response, because senescence, like what we talked about for the cells in culture, if you now stop that cell from dividing any longer, that's a really potent intrinsic anti-cancer mechanism. That cell will not be able to make now 2 cells or 4 cells or 8 cells. It's stopped at that point in time. It would be better if the cell would die entirely, so that then it's not hanging around. But maybe there's some benefit of having that senescent cell, that it will maybe trigger an immune response or something to then say, hey, something's not quite right in the system here. Maybe we want to activate some sort of response to this. And so that's maybe where the pathway was somewhat activated or induced to become senescent. And we see that with this particular model. Where we became very interested, and so in the context of why did we continue in this work, because it was very easy to make mice sick. And in fact, at the time when we were first shopping this story, we had frequently the comment of, you can mutate any one of hundreds of mouse genes and you can make a mouse sick. So what is the importance of this? Which is a valid point. What we were doing at the time was we knew that that mouse was missegregating its chromosomes. So we were doing what we had intended on doing with the design of the model, but it just didn't seemingly have enough time to develop tumors. So we asked a really simple, naive question of, there are lots of mouse models that have tumor suppressor loss that are prematurely prone to cancer development. If we now add chromosome instability on top of these mice that have a predisposition to cancer, is it much like what we see in our carcinogen treated experiments, where we see a synergistic effect and we get more cancers? Okay, so that was the simple idea of what we were doing. Because Jan doesn't like to do things in small scale, we threw a bunch of different tumor suppressors at this mouse at the same time. Many of them p53 knockout mice. P53 is an extremely important human tumor suppressor gene, mutated in lots of human cancers. P53 knockout mice, very tumor prone. P16 knockout, those animals also are tumor prone, not to the same degree as what we see for P53, but P16 is also a very commonly mutated tumor suppressor gene in human cancer. P21 is part of the P53 pathway. P53 engaged cell cycle arrest largely through activation of P21. Those mice also don't really have a really strong tumor phenotype, but we started asking that model as well as P19, which is upstream of modulating of P53. So essentially we have the P53 pathway based on P19, P53, and P21, and the P16 pathway, which both of them will inhibit different cell cycle regulators. And so the idea is that if you now are knocking out these important regulators, you don't have a stop of the cell cycle when it should be stopping, and you get transition through, and you can then proliferate very happily. Okay, so that was essentially the idea. What we found was actually quite remarkable, is that this mutant mouse model that we had that had a premature aging phenotype, it was quite severe. We could make it even worse. So if we knocked out the P53 pathway, essentially P19 or P53, we made the mice even sicker than what they were originally. Those mice also seemingly had a predisposition to tumors as well, which fit along the line of what we had anticipated related to the tumor development. It was a tumor study, was what we were doing. Interestingly, when we were looking at the P16 knocked out animals, if we looked at them for their tumor phenotype, it was a synergy where now we were getting cancers in those mice that didn't have P16, but they looked really healthy. So it was this very kind of dichotomous effect where these mice, it was a damned if you do or damned if you don't scenario, that if you had the ability to express P16, you had a very strong premature aging phenotype. If you didn't have the ability to express P16, you had a strong cancer phenotype, but they looked very healthy. So you were dying one way or the other, premature aging disease, or you're dying of the cancer disease, which led us to then the idea about this particular regulator. That was the only one of the group that I told you about that kind of had this differential effect, suggesting that the P16 expressing cells were playing a very important protection against cancer effect, but by their presence were actively promoting degenerative disease, which led to then essentially our next tool in the mouse model world of then saying, if these P16 expressing cells are so important for driving pathology, but yet so important for suppressing cancer, can we do something about those cells? Can we maybe manipulate that cell type so that it does, so it still keeps the ability to engage the arrest mechanism, but then we don't let it reside long-term within the organism to really promote the degenerative process.
Yeah, and so this has been a really valuable tool, and kind of, so you develop mice where you can selectively eliminate cells that are expressing P16.
Correct. So in my career, so we go back to the career, at the time, so I had just received my PhD from the university, from Radboud University in Nijmegen. And I was thinking very seriously about leaving Mayo Clinic at the time and going to the laboratory of Judy Campisi, an absolute giant in the field of cellular senescence related research. Interested, even though she herself is not a giant, but her work is very influential within the field. That she was at the Buck Institute looking still in the context about senescence and cancer, and she's done a lot of work related to why senescent cells have impact on other cells in the environment. Just an amazing individual that we unfortunately lost earlier this year. But so at the time, and I've always been very transparent with Mayo when I've been thinking about doing something different. And at the time was just when the Kogod Center on Aging was just starting at Mayo Clinic. That was started by Jim Kirkland, was kind of our first director of the Aging Center. And at the time, I was still having a bunch of cancer-related work, but there was a desire to have a program project-like grant that would be able to hit a number of different things within the Aging Center. That do we have the right type of ability to do something about senescent cells or not? And we thought, because we had this BUBBR1 mutant mouse model that had the premature aging phenotype, that we show that P16 expressing cells were important for driving pathology, but also preventing tumor genesis, we thought we had the tool to really test, can we do anything about that? And that was where we developed this ink attack model system. The ink attack model system, the ink comes from P16 ink 4A. That's the ink part of the gene. The second part is apoptosis through targeted activation of caspase, is what attack stands for. That had been previously shown with different promoters to be able to actively eliminate cells not via the typical way of having DNA chain incorporation through like HSV-TK mediated cell death. Because in the context of senescence, we knew molecules, or we thought molecules that were really important that we used for selection of embryonic stem cells in culture, where you have a lot of rapid division, DNA is replicating. So if you do something that then will stop that replication, you can promote cell death. Senescent cells are by definition really not cycling. So there's not theoretically much DNA incorporation. Judy's group has now suggested that the cell death mechanism via HSV-TK can happen through a mitochondrial mediated cell death, but we didn't really think about that as a potential at the time. So we were looking for a molecule that we could do a selective induction of apoptosis, which was this attack transgene. The attack transgene has a modified FKBP protein with caspase 8 that requires an artificial drug to be added for its dimerization and subsequent activation of caspase 8. That drug, AP20187, which I'll just call AP, I could fill you and I with as much AP as has been produced, and it will have no effect because we don't have the transgene. So it's not, it requires the genetic modification for there to be a benefit. And so what we had done, and this was, again, a crazy experiment, because we made transgenic founder mice that had the INK-ATTAC transgene. If you remember from your genetics approaches, when you make classical transgenic mouse models, every transgenic founder line is different than every other transgenic founder line. So line 1, line 2, line 3, they're all different because they have different incorporation sites, different number of transgenes, and so forth. We tested in vivo the ability of the INK-ATTAC transgene to have benefit on physiological function in the Bubber1 hypomorphic mouse model. So Bubber1, the mutant mouse model that I've told you about, itself is sterile. So we cannot interbreed these animals. It was a 1 in 4 shot of getting mice that had the right mutation to promote premature aging. Then you had half of those that had the attack transgene and half that did not. So then you're at 1 in 8 of mice that have this. We tested, I think, 11 different transgenic founder lines was what we had tested at the time. And what we found is that there were ultimately, in the context of the Bubber1 hypomorph, there were 2 transgenic founder lines that functioned. So you can look at this in one of 2 ways. We had 9 failures, if you want to look at it from the pessimistic point of view, where the transgene was not incorporated in the right spot, or it wasn't behaving in a P16 dependent manner, or we were maybe having killing in cells that we didn't anticipate having killing in cells. So a lot of weird stuff can happen when you're in the context of making transgenic mice. But 2 of the founder lines look like they behaved quite well in the context of the Bubber1 mutant mouse model, where we saw a similar benefit for what we had done with the genetic deletion of P16 by now removing P16 expressing cells with this drug, started early enough in life before pathology had come on. We were able to largely repeat the benefits that we had seen with the P16 knockout animals, where we had attenuation of disease, primarily of cataract development, skeletal muscle degeneration, and adipose tissue retention by now eliminating senescent cells.
And did you look at lifespan extension?
Good question. So in that experiment, when we looked at the hypomorph, we had not a strong improvement of overall lifespan, but the mice were not dying of cancer. Unlike the P16 knockout animals, we had no tumors that were developing. The phenotype that we think was largely killing this progeroid model, because I told you they died prematurely without a lot of cancer, relates to a cardiac arrhythmia effect. That the cardiac arrhythmia effect doesn't look like it's a senescent cell-dependent context. So in the context where we're now removing the P16 expressing cells, we really don't have a strong improvement of the cardiac function of that particular model.
Interesting. That's really interesting. Is anybody going to pursue what's going on in the heart?
We've done a little bit of work. It's interesting because this mouse model, even though we published the first report, I guess in 2004, so 20 plus years ago, I still work with the mouse model today that's still upstairs breeding. We're still finding out various things about it. But in terms of the cardiac phenotype, we haven't done a whole lot of work in that space specifically for the Bubber1 mutant at this point in time. We had an idea at a point in time that it looked like there was a mislocalization of connexin 43 that it was
Ah,
part of the ability to then have a synchronous signal that
interesting.
then
So
leads to all of
the gap junctions between the
cor-
cardiac cells.
correct. And that
Yeah.
connection 43 seems to be dependent on its phosphorylation that doesn't occur when BUBR1 is at low levels.
Huh.
The gene that we mutated, BUBR1, is a kinase. It's never been really defined of what are its particular substrates.
Huh.
But now science is getting much more improved in our techniques and technology versus where we were playing at the time for what were various kinase activities and so forth. So it might be an interesting thing to re-explore now that our tools are getting better.
You know, I know we're limited in time, so I don't want to take it, but in context of the brain, let's move to the brain and
Sure.
just follow up
So
on the astrocytes, which are the most prominent type of glial cell, equal in number to neurons. They're connected by gap junctions, so it would be interesting to look at those. But can you get to the brain?
sure.
So
So in the context of the brain, I'll tell a couple things. So related to the BUBR1 mutant mouse model, this prematurely aged mouse, we have found now with collaborators from our Mayo Clinic Florida group, that there seems to be a strong induction of senescence in endothelial cells that occur within the blood-brain barrier that cause disruption such that these mice have a lot of leakiness into their brains. If you remove their senescent cells, you see that there's, again, a large benefit, suggesting that the presence of senescent cells in the endothelium can promote dysfunction where now proteins that shouldn't be getting into the brain are getting there and vice versa. And we've also been then looking in the context of normative aging. So this INCATAC model, we had the comment that it was very easy to make mice sick. What relevance does this have for a normal mouse? We did do an overall lifespan experiment on normally otherwise wild type mice of doing the same treatment paradigm of treating prior to disease onset. And what we could find there is that many diseases associated with age that occur within a laboratory mouse can be prevented with senescent cell elimination. There's also an improvement of median lifespan in those animals. I think we didn't have enough to really make a judgment call on the maximum lifespan extension. We didn't do it quite right. But I also told you that we started with 2 transgenic lines. One of the 2 lines, if we were looking at male wild type mice, for whatever reason, those mice had the insertion somewhere really negatively, that in the absence of treatment, they were dying after about 15 months of age. Something we didn't see in our hypomorphic mice because they never get to 15 months of age. They're dead by 12 months of age. And so that line has some significant impact that we can't prevent.
yeah.
There was some improvement of lifespan with those models, but then we've now been basically eliminated down to 1 founder line from the 11 where we started from. So that's the INCATAC model that everyone is using, is this one particular founder line that we ended up doing the in vivo testing of. At the time, so I was going through my career, I was doing my postdoc, making this INCATAC model, seemingly was turning out. And I was intending to, for a variety of familial reasons, was trying to maybe stay at Mayo Clinic. So again, being very transparent, my mentor was still here at Mayo, and we made a very simple delineation of saying, I guess my lab and what I'll focus on is what I call neck up biology, and he'll work on neck down biology. Okay, so that was my entrance into brain ponderings, was I was taking the neck up because I was also very interested in the cataract phenotype that we had in these mice. Unfortunately, funding agencies are not so interested in the molecular mechanism behind cataracts, which I have a really good understanding for. Maybe we'll be able to revisit that at some point in time, but that was why I got into neurodegenerative research, was really trying to find something of a tool because we had this really rapidly aging mouse model. I wanted to use tools that were also rapid disease models of neuropathology to now see, are there senescent cells relevant for these particular diseases? So my lab, I came at it very naively, I think, and I think I'm still very naive in the space, because we were looking at Alzheimer's disease. I've talked with Ron Peterson, who's here at Mayo many different times, and I was looking at AD pathology and essentially separating it into the 2 main pillars of whether it's an amyloid-based model or a tau-based model of disease, instead of putting them together. And so we had done a couple different experiments where we had bred the INCATAC model onto some more amyloid-based models of disease, or to a tau-based model of disease. And we'll talk primarily about the tau-based model of disease for the remainder of this, because that's where we have some more information. Because to this point in our hands, we don't see a very strong induction of senescence in the amyloid alone models in our hands. Maybe we don't have the right ones, and that's the problem with the amyloid model, with any of the models that we're doing for mouse model neurodegenerative research, is that they can behave somewhat routinely, repeatedly in some places, and maybe not so repeatedly in other places. But we didn't have, when we got tissue from other places that had done more amyloid alone models, we didn't see strong upregulation of senescence-related changes at the time related to P16 upregulation, factors that are involved in the senescence-associated secretory phenotype, the pro-inflammatory signaling occurring from senescent cells. So we focused very much on this tau-dependent model of disease. The mouse that we use is the PS19 strain that has a P301S mutation, developed by Virginia Lee and first published, I guess, in 2007. I was somewhat impatient based on, starting a lab and wanted to have phenotypes. So those mice get pathology really very aggressively and quickly. And so we did an experiment where we crossed the Incotact model onto the PS19 mice, treating them prior to disease onset. So this is not relevant for people that are coming into the clinic here, because you're not coming in prior to disease. You're coming in with some sort of ailment that then you're wanting to have some treatment. What we found in that model is that it's very important, the timing of treatment, which is not surprising, right? In the context of neurodegenerative disease, much like cancer. So if you make still the same analogy, is it better to treat a cancer when it's a stage 1 or a stage 4? Well, your outcomes are probably much better at stage 1 than it is at stage 4. Same with neurodegenerative disease, that how much of an ability does the brain have intrinsically to repair these processes once the scar or damage has occurred seems to be not terribly awesome. But if you can kind of change trajectory, that's where we've had some benefit. So what we were doing was we bred this Incotact model onto the PS19 strain, treating mice to remove their senescent cells prior to disease onset, just to see whether we had an impact or an improvement overall in their pathology. What we found is that we did. And so then, okay, why? Now we need to figure out what is actually happening. We saw that we could really negatively influence, so there was a senescent cell induction. What we were trying to then figure out was, what is the origin of the senescent state in this PS19 strain? Naively, we utilized SA beta-galactosidase staining, senescence-associated beta-galactosidase, something that was first published about 30 years ago from Judy Campisi's lab, of a notice that senescent cells change their lysosomal activity to be very active at a particular point in time. We coupled that, because of work that we had done in the context of normative aging. We had done beta-galactosidase staining followed by electron microscopy to look for where there were the inclusions of the beta-gal crystals, to now look at cell morphology to give us a bit of a hint of what cells are now having the beta-galactosidase expression. Because in the space, our tools are still not awesome for senescent cell research. And if you were to ask me today, what is the best marker, I can't give that to you, because I think it's very dependent on very different contexts. So in the brains of these PS19 mice, what we found is that there seemed to be just a lot of background neuronal beta-gal activity that wasn't changed.
We found that too. Yeah.
So maybe I should be asking you questions coming up here.
No.
But we found.
I mean, we followed you, right?
Yeah. But what we found, but that was not changed with our treatment. So that background that was happening within neurons didn't really change with our Incotact mediated elimination strategy. We found that there were other cell types, primarily cells that look like astrocytes and look like microglia, that had the inclusion of these X-gal crystals, that we then were doing our treatment paradigm. So unlike where you maybe have a drug or something, we have this genetic model where we can remove P16 expressing cells. And we saw that there were fewer of these microglia that had the X-gal crystals and fewer astrocytes that had X-gal crystals, both of them in the model. The ratio that we had for numbers overall, we had probably about 3 times as many astrocytes that had the X-gal inclusions as microglia, but I can't tell you which one begets which, or if there is any relationship between those two. So that's
And there are a lot more astrocytes than there are microglia.
and so maybe it's just a numbers thing, that because there are so many astrocytes, that's why there's threefold more. But what we've been doing since that point in time, so in work that's currently unpublished, is now we're trying to convince the brains of these PS19 mice in a cell type specific manner to be better or worse in a senescent cell context. Meaning we're trying to make more astrocytes senescent in the PS19 mice or less astrocytes senescent in the PS19 case. Interestingly, the model doesn't care about how many senescent astrocytes it has. The pathology is consistent. So if there's many more, doesn't care. Less, doesn't care. Pathology is identical. Microglia, on the other hand, that it cares about. So if we push the dial on one side of making more senescent microglia, pathology is worse. If we prevent senescent cell expression specifically in microglia of the PS19 animals, there's an improvement.
Now, it's been known for a long time that there's what are called activated microglia associated with the pathology in Alzheimer's. And it's also well known that these activated microglia produce a lot of pro-inflammatory cytokines that are thought to contribute. So what distinguishes that phenotype from, a cell that has a microglia that has senescence markers?
Great question. And we thought we would be very, very smooth in answering that question of doing something in vitro, of having microglia that were resting, activated microglia, and then we'd convince microglia to become senescent. And we just Venn diagram that and then say what was unique about the one versus the other and so forth. In vitro, we were extremely efficient at making reactive microglia. That we had no problem doing. But anything that we tried to do to convince them to not be so stressed out didn't really work. So we had no node of the resting state. And when we were trying to then stimulate them to go into a senescent process, they were dying, was the consequence. So in vitro, we couldn't really do that. We resorted recently to just doing a microglial isolation from the PS19 mice to then pull out the microglia from the brain, and we found the three populations we would have anticipated. So we wasted a ton of time and energy in an in vitro approach that we then were able to answer with some in vivo, because technique and technology has improved such that we can do single cell profiling of the microglia that are coming from these animals to then look for these distinct states. What we found, and what I think is somewhat in agreement with others, but there's different tools, different mouse models are probably behaving somewhat differently. What we found is that the senescent population in the PS19 mice was underneath the overarching umbrella of the disease-associated microglia. So there are many different subcategories of disease-associated microglia in this PS19 mice. There seems to be one particular group by single cell profiling that looks to be much more senescent or having markers that look like different pro-inflammatory signaling molecules, but some of those are going to be in agreement with a lot of the disease-associated microglia or the activated state of microglia as well, which in the senescence-associated secretory phenotype area, the SASP, there's many of those phenotypes that look like they're just part of the activated state of microglia. So it's kind of this new transition state where they've gone into the disease-associated microglia. Something that maybe you can explain to me is that it looks like maybe within that population, if we do pseudotime analysis, it looks like the senescent state for the disease-associated microglia has first gone through something where it tried to proliferate, or there was a proliferation signal that then maybe there's a blockage or something that goes awry in those cells to then trigger them to go into a senescent state, or maybe the induction of senescence itself, or the induction of proliferation specifically of these activated disease-associated microglia stimulates them to transition into this different state. Something we're still kind of exploring at this point.
Off the top of my head, I can't, think of what to do next with that particular question, but and
so
and then
work from others, not us, has done some interesting studies where they've done co-culturing of neurons with neurofibrillary tangles. So our model theoretically is having disease in the neuron. the neurons, we don't have real strong evidence that they're becoming senescent. We have cells in the microenvironment that are becoming senescent. So why? What is that relationship? We're still working on that in terms of what are the potential cues coming from neurons that are stimulating this.
I do have an idea.
Okay.
Now that stimulates them. We did all this work beginning in the 90s, early 90s, showing that in neurons in Alzheimer's disease, that affected in Alzheimer's disease, and we used post-mortem patient tissue, and then we also exposed cultured neurons to oxidative stress, amyloid beta peptide. That two things. One, there's a lot of membrane lipid peroxidation occurring and release of a toxic aldehyde. It's a little bit of the phospholipid that's cleaved off by free radical attack. And it can covalently modify proteins on lysine, histidine, and cysteine residues and affect their function. So one experiment I would do is just, you can buy 4-hydroxynonenal and just dump it into microglia cultures. And then another thing we found was there's a accumulation of long chain ceramides.
Yeah.
And, so these are a couple of things that have, let's say, might have a effect on microglia that pushes them maybe towards a senescence phenotype. That's just two things off the top of my head.
And so work from not us, but from Ria Grazia Spillantini's lab has shown an idea about the idea of having neurons that have neurofibrillary tangles being co-cultured with microglia, that those neurons have the eat me signal such that the microglia will go and they'll phagocytose the neuron. The problem is that those particular neurons don't seemingly become destroyed within the microglia, and the microglia then changes its behavior to then say, holy cow, now I have a problem of I can't do what I was supposed to do. And looks like when it goes into that state, starts to secrete many of the factors that we think about of the senescent state. The cell, the microglia, then loses its ability to phagocytose any more neurons, so it kind of becomes stuck and now activates that signal, which would be basically in agreement with our model, where we have the neurons with neurofibrillary tangles, the microglia, they maybe engulf them, become senescent. If we then go in and eliminate them, then you remove this negative signal that's occurring. But then, it's at least conceptually the idea of what we're kind of working with right now.
Huh. That's really intriguing. And so you made these mice where you can selectively remove cells with a senescent phenotype, and you see this normal aging, and then in pathologies associated with multiple organ systems, including the brain, one, accumulation of senescent cells, and two, if you remove them, beneficial effects. You focusing on the brain and your mentor focusing on everything down. Okay, now there's a big effort and even companies being developed around developing chemicals, drugs, if you will, that can remove senescent cells, or I guess maybe otherwise modify them. These are referred to variously as senolytics or Senotherapeutics. To finish up, can you talk a little bit about that? Because there's even clinical trials going on in certain disease conditions.
Correct. So in the context of the company development, we were involved with a company that formed that's now known as Unity Biotechnology, that it still is in existence in California is where they're at. But the company, when it started, some of the scientific co-founders were my mentor, Jan van Dersen, Judy Campisi was another of the founders, and Dao Hong Zhu is the third one. And what we were doing at the time when the company formed, I would not advise as a great way to start a company, when we didn't really have a drug, nor did we have a disease. We had an idea about that senescence was bad for you, largely based on some of our strategies where we had removed senescent cells. So since that point in time, we had shown that it was safe and effective to remove senescent cells from mice, that they weren't keeling over dying. many people here, the first time that we started doing our treatment in the INCATAC model system was that they anticipated that we were just going to have all of our mice turning up dead because maybe the transgene was on in the neurons, or something where there could have been a lot of negative effect. But we don't see that. There's not a real long-term negative impact with our genetic strategies of eliminating senescent cells, which gave credence to the idea of, well, let's now look for agents that can do this. Jim Kirkland was one of the people that was pioneering in this space related to looking for what is really different about a senescent cell that might be a space that can be intervened with. And what they were looking at was something called senescent cell anti-apoptotic pathways, or these SCAP pathways, where senescent cells have a high upregulation of many different pathways that are actively keeping the senescent cell alive. We used to think about senescent cell biology, at least I used to think about senescent cell biology, of the analogy that they would exist in a valley, and it was going to be very challenging to convince them to behave differently than what they were, that they were very stable. And convincing them to move out of that or to die after that point in time was going to be very challenging because of all of these pathways that we're on. What seems to be repeatedly now is that senescent cells are much more teetering on the cliff of destruction, more so than that they're very stable, because many different players that we have that can now manipulate some of these certain pathways seemingly are sufficient on their own to promote death, because there's so many things that are apparently going on within that cell. What my lab has been focusing on primarily is molecules that are involved in inhibiting BCL2 factors of anti-apoptotic pathway. The negative consequence is that there are certain cell types within our body that are reliant on the same pathway for their survival. So there are off-target negative, I say off-target, but it's completely on target. It's doing what is designed to do in a senescent cell, but there are normal cells that have the same pathway required for their survival. So that's an effect that is not desirable, but it's very much on target for what the molecule is doing. Things like navitoclax is the molecule that we use very frequently. There's other molecules, a combination of dasatinib and quercetin, which you yourself have published on, related to how there seems to be a benefit for various mouse models of pathology with dasatinib and quercetin treatment in mice. And that was based off of Jim Kirkland's stuff related to pathways that look like we're being able to be targeted by molecules that we already had on the shelf, such that we could get them into
and have already been used in humans. In
Correct.
fact, the dasatinib is used for
many of these molecules. So your idea about Senotherapeutics is, I use, again, I like analogies, that I use the analogy about Senotherapeutics that senescent cells are a tree that have all kinds of different branches of different inflammatory signaling or whatever is going on within the tree. With a senolytic approach, with our genetic approach, we're cutting the tree off at the base, that we don't want to have anything that's going on from that senescent cell, whatever the pathways different are. The xenomorphic strategies, those are abilities to try to take off particular branches, that if you think that this is the branch that's really driving pathology, let's only take away that and leave the rest of the tree intact, maybe a bonsai type effect, maybe if you're thinking of it that way, that then there's still healthiness after you've taken away the diseased tissue, something like that. So that's the analogy I think about senolytics versus xenomorphics. Largely, much of our work is focused on the senolytic strategy because of our genetics. Our genetic approaches that we've been using are taking the tree off at the base. So that's where we're coming with our senolytic strategies. In the context of companies, so Unity was one. Many others are playing in the space now. So Unity, since that point in time, has now developed molecules. So we have now a way to eliminate senescent cells. Dasatinib and quercetin is one, but there's also proprietary molecules that are being used in different cases, as well as different disease conditions. The first clinical trial that was done from Unity was related to osteoarthritis, was what it was. And in that particular case, unfortunately, the clinical trial did not yield positive result. And so that had a pretty significant impact on the company and kind of changed what they were working on. Now what they're working on is really quite exciting research on diabetic macular edema, where this is a condition that diabetics get within their eye. They've developed molecules that are injections into the eye, such that it's a local administration, because they think there's endothelial cell senescence that's occurring within these particular patients. When that happens, there's a lot of reorganization of the endothelial network within the eye, leading to a lot of problems. such that if you do this treatment, you kill off the senescent cells. There's not very many of them, but by killing off those endothelial cells, there seems to be a replenishment with normal cells, such that those patients, the first clinical trials have been published, that if you look at DME patients that are treated with vehicle over the period of a year, so a one-time administration of the senolytic drug, patients that receive the vehicle, you have glasses on, I have glasses on, so we've done vision tests. What diabetic macular edema patients typically have is a loss of 2 lines of text over a year, meaning that they have to move up 2 lines on the chart to see the letters. These patients that have received the senolytic, it's stable, so they do not lose their vision over 2 years. So pretty exciting work related to a specific patient population with a specific agent with a very targeted delivery.
And that's one point, even in the animal studies, right? The treatment doesn't have to be continuous. It can be, intermittent. Or one last thing.
Well, much of our work, so we can go over, it's fine. So much of my work,
okay, good.
we've been more working in the space of, we want to see biological effect. So I'm sure we're overdosing our mice. We haven't done the intermittent kind of strategies that would be relevant for the clinic. Our patients, our mice upstairs, are very compliant with their treatment regimens. We don't see off-target toxicity with the molecules that we're administering. So we do pretty much consistent treatment to really change the trajectory as much as we possibly can. What we found, and what, related to some of the unpublished work, because it's not terribly beautiful, is that in the context of our degeneration model, there is a point of no return, where if we're treating mice that are too diseased, they don't care about whether the senescent cells are removed or not, because the pathology is already severe enough that it has no ability to restart. There's maybe ideas that you can think about by now, coupling ideas of cleaning the soil if there's contamination of senescent cells, to then maybe have a reactivation of maybe various factors like Yamanaka factors, if you're thinking about doing reprogramming, to now try to stimulate those cells that are remaining, that are the healthy cells, to now have ability to divide or replenish or something at that point in time. So there's all kinds of things that you can think about that could be really fun to do. It's just that we don't have the type of money that would be required to do all of these experiments. And that's why we haven't done a lot of the intermittent type of treatment. But with that clinical trial from Unity, that shows clearly that if you're on target for the disease-causing etiology and you can prevent that, there's pretty significant impact. In the context of AD pathology, there is a relatively well-known clinical trial going on right now with dasatinib and quercetin, trying to also really change trajectory of disease course in people with mild cognitive impairment, for example. Of these molecules are relatively safe. They don't seem to have they seem to be well tolerated. And that's not work that I'm directly involved in, but obviously following the literature in that space. So it'll be interesting to see, because those molecules, dasatinib and quercetin, or other, even much of our senolytics, are molecules that can have on-target senescent cell killing ability regardless of where they're occurring. Even a strategy of our genetic approach is also, if it's developing senescent cells in the liver, we're killing the senescent cells in the liver as well as in the brain. And with the interorgan communication that's occurring about how senescent cells can be having really distal impact, you're changing phenotype in many different tissues, such that then maybe overall you have a dampening of the signature of senescence by removing the senescent cells that might have a benefit for the patient. And that's ultimately what we're looking for, is improvements for the patient. We'll have to figure out mechanistically why we see those improvements in the end, if we end up having improvements. But those are some of the caveats with our current approaches for intervening with the senescent cell phenotype.
Yeah, the one thing you've mentioned several times is, in the animal models, and it's the way it is as far as clinical trials in Alzheimer's disease. The current kind of consensus is that by the time someone is so symptomatic and diagnosed with probable Alzheimer's disease, there's so much pathology that it may not be possible actually to completely halt the process, or probably definitely not reverse it.
Yep.
So the idea is to have early diagnosis, and Ron Peterson, your colleague up there, has been kind of a pioneer in establishing criteria for mild cognitive impairment. And so a lot of the clinical trials now are kind of going to that stage. Now,
Yep.
I've been interested, as you may know, for a long time in how can we reduce someone's risk?
Yeah.
And epidemiologically, well, there's kind of three things that seem to stick out and are supported by studies in animal models, for example. Physical exercise, regular exercise, not overeating, and keeping your mind intellectually challenged.
Mm-hmm.
You've done some work, actually. We did a study a long time ago where we had evidence that daily caloric restriction and intermittent fasting can be beneficial in an Alzheimer's model if we started fairly early, actually, before there's hardly any pathology in this 3x TGAD model. And you followed up, and you've also done, you did a calorie restriction study too, right?
We did, yeah. So in the context of senescent cells, anytime that I give a talk, then people say, well, what should I do now? What can I do today? Diet, exercise. You already hit on two of the three related to things that we can modify that we know healthy living has an impact on, that we see that there are fewer senescent cells that are accumulating. The caloric restriction study that we did just recently, the concept behind it was not exactly a caloric restriction study. The concept behind it was actually a hibernation idea, was what we were trying to do. So with colleagues from the Netherlands, what they have seen is that in hibernating species, many of them mammals, bears, and other sorts of things, possums, ground squirrels, that if you look at their brains as they're entering into the state of torpor or their state of hibernation, they have a very high upregulation of phosphorylation of tau. Okay, so their neurons become very hyperphosphorylated with tau when they're going into the spot of slumber. When they're coming out of this torpor state, that goes away. So it's a very reversible phosphorylation state of tau, which is unlike what we see within pathology. So with AD pathology, it phosphorylates and it stays phosphorylated. So there's probably different kinases and phosphatases that are involved in this process. And so what we were then thinking was that if we can convince laboratory mice to go into hibernation, which this collaborator had done, it was through a pretty significant caloric restriction paradigm, we can then induce the phosphorylation of tau. And now when they're coming out of it, maybe we can have a prevention of that. And we're doing that in a disease model. So they have the point mutation of tau. And is there now a particular kinase or phosphatase that is activated as they're coming out of slumber that then we can potentially utilize as a new approach for intervening with tau dynamics in pathology, was kind of the concept of the study.
That's intriguing, because it turns out tau is this microtubule-associated protein is hyperphosphorylated during early brain development, right? When there's a lot, the axons, dendrites, well, it's mainly axons, where tau is. But anyway, they're rapidly growing, synapses are forming. So it's interesting that in states of kind of normal plasticity, healthy plasticity, tau can be hyperphosphorylated, but it's also hyperphosphorylated in the neurofibrillary tangles.
Yep. And so what we had done then was we were trying to convince these mice to go into hibernation, but their hibernation protocol is on a daily cycle. So they go into slumber for about 21 hours, and then they're awake for 3 hours, and then they go back into slumber. And the way that we're doing that is by influencing when they can eat their calories, is the idea. So what we had done was we had done this in our tau mouse to then see whether or not there was a change of, now, could we change the pathology? What we found is that there was a clear impact on their behavior. So there were behavioral improvements in these mice. That was not just because they were more active wanting to eat during the time, that it was actually something that we saw by mazes and other things for the mice, that they were behaving as if their cognition was improved. But if we looked at the overall neuropathology of these mice, there was not really significant changes. It was kind of a situation where we could improve phenotype and behavior without really changing underlying biology. But part of that looked like there maybe was a bit of a stimulation of some adult neurogenesis maybe that was going on in the model. But it was a system that we were working with a shared MD PhD student that she then came here for a period of time and then returned back to the Netherlands to finish her work. But that was where the concept was very solid, but then overall the results were a little bit hard to interpret, I think.
It's interesting though that these, so bats, right? Bats also have the state of torpor, right? And they go, and bats live a lot longer than mice, even though, some species of bats are the same size as a mouse.
Yep.
And there's some thinking that this cycling in between torpor and non-torpor.
Well, and you also think about the energy expenditure of bats, of how that they have to go out at night, then they're hunting bugs, that they have to have good echolocation, that their ears need to be completely functioning. Otherwise, they're also not going to find their prey that they're needing to eat. And the maintenance of that system is very robust. If you look at those animals being able to live 2, 3, 4 decades, even in the wild, relative to, we're not having mice living that long in the wild, I'm pretty sure.
Yeah. Okay, so what's going on now? I mean, you've done a lot. You've been really a leader in this area, and I'm kind of want to know what's going on now that you can mention without
getting myself in trouble. So to me, I think something that is still surprising to me is how common the senescence node is in promoting dysfunction in different pathologies, including within the brain. Because you look at different disease models, whether it's a Parkinson's model or an ALS model or an AD model or a tau model or an APP model, you have different cell types, and you've published this too, that in your model, you seemingly find a senescent cell context, but you're not finding microglia and astrocytes that look like they're becoming senescent. The precursor oligodendrocyte cells look like they're the ones that are becoming senescent. But yet that node of involvement really promotes dysfunction such that if you're intervening in it, you can have many improvements. I'm still shocked that that is the case, regardless of what is the tool or the model. So I don't think that you're wrong and that I'm right, or that I'm wrong and you're. I think that the tools are kind of suggestive that it's going through this particular path in your condition, and it looks like it's actively promoting dysfunction. So that's to me still somewhat surprising. So lots of work is being done in the space still related to how relevant are these cells in the context of normative brain aging, normative cognitive behavior. I've kind of steered myself a little bit away from that area, largely because I don't want to be competing very actively with others here at Mayo. So there's a number of different researchers that are working in the normal spaces of cognitive aging to see whether or not senescent cells are there. And largely, it looks like they are as well. And maybe it's different cell types, but it also kind of has hints of microglial involvement of senescence as well. Within my program, what we try to really do is develop new tools, new approaches, new technique, where we can really identify and promote dysfunction within particular cell types. I mentioned driving senescent pathology in particular cell types to then say, how critical is that one cell for driving the pathology or not? If we can just intervene alone in that sample, is that sufficient? Because then that might be our next generation of approaches for targeting intervention, where we're now not hitting all of the other senescent cells about what might be a beneficial state. Let's only go after the ones that are really responsible for the pathology. Or if we're actively promoting apoptotic death of those senescent cells alone, does that have disease modifying effects or not? So those are some of the things that we're working on within my laboratory right now. And I guess it's a to be determined or stay tuned kind of approach.
That's a very exciting goal, and I look forward to seeing how that turns out. One thing I want to mention to you, it might be useful, you may already know about it. Before I retired, a neurologist under me, Dimitros Kapagiannis, developed methods to, all cells secrete these small vesicles. They're called exosomes or extracellular vesicles. And using an antibody that's fairly specific for protein on neurons, he pulled out of the blood extracellular vesicles that we think are coming from neurons, and then he molecularly interrogated them. It would be interesting if you could find some you know, it's called the senescence associated secretory phenotype. These
Yep.
senescent cells are undoubtedly pumping out a lot of these vesicles.
Correct.
Some of them, even from the brain, but from anywhere, may get into the blood. And it would be interesting to identify like a blood-based biomarker of senescence.
Yep. Absolutely. We've been looking a little bit in that space, and you're right. There are a number of these extracellular vesicles or exosomes or all kinds of different things that are secreted that senescent cells seemingly do. What we haven't had a really good tool for is looking at that on a cell type specific manner. So then identifying which ones are coming specifically from a microglia, for example, that might be senescent and so forth. So tools are getting better. All of our approaches are getting better. What we used to think was, cutting edge about microarray technology. Nobody does microarrays anymore. It's single cell RNA-seq, and then you can easily identify where these populations occur, which we would have not had the right tools for doing that a decade ago. And so it's just moving so fast that you kind of have to keep up with what is changing to really understand and identify what some of these factors are. With our tau model of disease, what we were trying to do was really characterize, because the strain is good, but there is also a variability of penetrance of the phenotype, much like happens in many mouse models, where we can have some 6-month-old mice that are very diseased and others that are really not diseased even at 11 months of age. And so why is that? So we were looking for tau depositions in the retina of these mice was what we were trying to do, to then stratify those that are now having aggregation of tau within the retina to now say that those are probably the most diseased. Let's intervene on those animals relative to those that are maybe more mild to kind of stratify populations. So a different strategy that is also not terribly invasive, because you could do the imaging with just having the animals being asleep and not needing to do anything else. But blood-based markers of senescence in the blood, a lot of that work is being done very actively right now, and including by, a lot of people from the NIA are doing a lot of that work.
Yeah. Very good. Darren, it was a pleasure to see you again. It's been a long time you came to NIA. I don't remember when that was, but
that was when I was very seriously thinking about leaving Mayo Clinic for the NIA in 2016, I think was when that was. So
that's right. Yeah, and Mayo Clinic's a great place. As you know, I'm from Rochester. I was born in St. Mary's Hospital, which is the main hospital for
same place I was treated when I was a kid. So.
Yeah.
Yeah.
Yeah. So it's great to see you, and I appreciate you taking the time.
It's always great to talk to you, Mark. And if you need a part 2, I'm always happy to come back too.
Okay, we may do that.
All right. Sounds great. Have a
Take
good
care.
one.
Yeah,
Talk to you soon.
bye.